I think a thorough introduction to the field, important directions in clinical research now, some fundamentals about models, available systems that we use in the labs, and there are 2 microphones, and so we have time for questions to be directed at anyone in the group here.
Hi. David Bates from the University of Nottingham. Can I just ask some questions about the PDX lines? We've been doing the same sort of thing in Nottingham for the last year or so. And the group has been doing lots of different tumours, from GI tumours, lung cancers, and so on. And generally, it's pretty tough to grow tumours in mice. And we're pretty astonished that actually the cholangiomas are probably the easiest tumours we've ever had to grow in mice. We get about 80% of our tumours take and grow, and then can be passaged. Do you have a similar experience? And any thoughts about why these particular tumours are? I mean, I'm not complaining, but any reason why you think they're particularly good at growing? Are they just particularly aggressive, or is there something about the immune response or something that makes them so easy to grow?
So we certainly don't have as high a rate as that, although we're doing a lot of biopsies as well. Is this microphone on? Yes. So that is higher than certainly what we're seeing. Perhaps your subset might be more aggressive. Certainly we found that the more aggressive cancers in the patients are more likely to grow and to grow more rapidly. But I can't say I can speculate on that, because we also do other cancer types. And for example, it seems like pancreatic cancers are somewhat easier for us at our end. So perhaps some nuances of your technique.
Yeah, pancreatic cancers are definitely easier than most of the others, but the cholangiomas are really, really easy.
Yes, sorry.
Yeah, actually this is great to have such a breadth of different types of talks to sort of integrate thoughts. One of my thoughts is there has been some emphasis on utilizing molecular or stromal targeted therapies together, or molecular or checkpoint or immunologic therapies together. What are your thoughts on how best to model how genetic hits might alter stromal biology, and how would we best process going through sort of screening different approaches in the multiple models that are available for looking at targeted therapy and immunotherapy interaction, or stromal and targeted therapy interactions? What are your thoughts on that? Wide question, sorry.
I can give a brief answer, is that from what we've seen for other cancer types, the kind of comprehensive modeling of the genetic subtypes has revealed a lot about the sort of different character of stroma. So, and largely this seems to have been borne out when looking back at human specimens. So the beauty of these mouse models, when they accurately recapitulate the histopathology, they have a very defined genetics. So it's often easier to see a signal for a unique change in a molecular signature, or potentially a stromal microenvironment. So I'm a big proponent of a kind of comprehensive genetically engineered mouse modeling, but I'd like to hear from the rest of the group.
Okay. Katie or Andrew, maybe you have thoughts on that?
I think modeling the complexities, all the different parts of a human microenvironment are very difficult, and I certainly defer to Aram and Nabeel on their input on that. But I definitely think looking at the trials and development and the results in immunotherapy, and particularly the adoptive cell therapies, it highlights the importance of looking at all parts of these models together. So it's a great question, but I definitely defer to the cell line models and the animal modelers to answer better.
Yeah, I think that's a focus or an interest. I mean, most of the models that we've worked on are KRAS driven, so they probably have a similar immune phenotype, maybe somewhat like pancreatic cancer. But I think looking at other combinations, maybe via viral delivery, and you don't need large numbers of tumors to discern a very strong pattern. So that could be a very efficient way to look at the impact of some of these other combinations. A lot of the models, even the IDH1 model right now, is, at least the mouse model, has a KRAS component, so it would be nice to try to find find other combinations that give good in vivo phenotypes. I don't know if that makes sense.
I guess I'd also like to add, just as a clinical researcher, it highlights the importance of early biopsy studies to make
Yeah.
sure that the effect we're expecting to see is actually happening in the human microenvironment, to see if we're on track with what the animal models show.
Other,
Well, perhaps it's not related to your specific question. I think as a clinical trialist, I think what's really haunting us is the predictability of each of the model. Which one will give us a better sense that will actually predict the clinical trial success? I think when you try to model things to answer more questions, I think the model may be actually more difficult to recapitulate, the specific pathway. It may actually have the limitation instead of the advantage. Yeah.
wanna go ahead?
Thanks very much, everybody. Excellent start. We have a reasonable idea. I think we would say there are probably around 6, maybe 7 targets in 2017, clinical targets with clinically potentially effective agents. But often the way we get there is a bit piecemeal. You have a test for this, you have a test for that. How close are we, and perhaps Andrew's probably best, how close are we to a sort of bespoke cholangiocarcinoma test you can do up front?
Can you just, John, can you just repeat the last phrase? Because some noise.
How close do you think we are to a bespoke cholangiocarcinoma test? One test.
So
just sign.
this is actually an excellent question. As a clinician, I always reflect this one on a daily basis because I actually make the effort, trying to get the test sent, get the test done, and also I'm responsible to interpret the results and to guide my clinical decision. I have to say, right now, 80% of the time, I still think this is probably just a routine exercise. And at the end of the 2 weeks, 3 week time period, the information is totally not very informative to me and also to the patient I'm caring for. But having said that, I also reflect on the other side, and that is, for this population, the treatment options are so limited. I feel like, I own these patients, every single bit of hope, and the hope doesn't come from random trying. I think, I think Katie probably can comment on the predictability of immune therapy, where there, you may have some insight for certain drugs. But for these target agents, if you don't actually perform the molecular testing, you really have no way guiding your patients for the next treatment decision, either on trial or even off-label use. So once I settled on that, I actually use that more often. And luckily, in my institution, we actually have a pretty reasonable system to get that executed and also to get that reimbursed. Yeah.
Thanks. Can we, want to go ahead?
Yes. I have a question. My husband has bile duct cancer, which has more than likely originated from a liver disease that he has developed since he was a baby, which has come from he's missing a chromosome. My questions are we anywhere closer to replacing these chromosomes, and how they affect trials?
I don't know a way yet to replace whole chromosomes, but with regards to trials, do you want to talk about? I think it would depend on the mutation, but
I think it would definitely depend on the specific genetic defect. Certain hereditary cholangiocarcinomas arise from a BRCA1 or 2 mutation, or microsatellite instability, mismatch repair deficiency. Something, a congenital liver disorder, may be a very different pathway. So I think we'd want to look at the specifics of your husband's case, but there are certain hereditary types of cancer that may have a different treatment path depending on the fundamental defect, whether it be DNA repair, which may be responsive to a PARP inhibitor, which are in clinical trials right now, or mismatch repair, where we know that that's probably the best biomarker currently for immunotherapy. But again, it would depend on the specifics.
thank you. Dr. Gores.
If you follow the hepatocellular carcinoma literature, we now understand that there's tremendous intratumoral heterogeneity within nodules with multiple branch points. What do we know about that phenomenon in cholangiocarcinoma
Are you talking about intra or intergrade?
within the same nodule?
So this is a fascinating question, Greg. We've been actually entertaining that, when we actually subject our patients on the BGJ trial. I think Lipika is going to discuss the data a little bit to shed the light on this question. There's no doubt both the intra and intertumor heterogeneity do exist in intrahepatic cholangio, and some of them may actually contribute, in our opinion, to the drug resistance mechanism. We just don't have a very established data to prove that. But having said that, I think we're wide open understanding this one. I think we're setting up some system trying to get multiple biopsy within the same tumor to answer your question. Yeah.
I do have one small comment on that. Is that in the TCGA paper that will be coming out quite soon in intrahepatic cholangiocarcinoma, the allele frequency for the very common mutations that you've heard about here are all quite high and comparable to each other, as though there's a common clone which has the ARID mutation and the KRAS and so on and the IDH mutations. So at least for these well-established drivers, they seem to be probably early events and uniform in the tumor. But there might be other more subtle heterogeneity, I think.
Yeah, but the secondary mutations, if you will, may actually turn out to be the drivers.
Yes.
I think one last question quickly, and then we'll make sure we have time for a break.
Yeah, hi, it's Altabdar from CPMC, California, San Francisco. I have two questions. We are doing PDX almost in 9 cancers. So we have 80% tumor take in melanoma. But coming back to liver and cholangiocarcinoma, we are kind of struggling, especially in liver. So my question is, do you do the PDXs in orthotopic or subQ? And the second question is, Dr. Andrew, the CTC, the allele frequency, how confident are you that the allele frequency which you are seeing in the CTC are exactly comparable if you do it in the tissues?
So for the first question, one of our collaborators is doing orthotopic PDXs, and we've been doing subcutaneous. And it looks like the success is good early on for the orthotopic, a lot more work. I can't say whether it's going to be better. The gentleman who mentioned earlier, perhaps he can comment on whether he's referring to extrahepatic, because extrahepatic and cholangio and pancreatic cancer have quite similar biology, and our sense is that those have a very particularly high take compared to intrahepatic. But
Thanks. We don't see a difference between intra and extrahepatic. I think we're doing subcutaneous models, and I think it may depend on the mouse strain line.
Just to answer your question very briefly, with regard to the CTC versus tumor, we don't have a lot of experience for intrahepatic cholangio at this point. We're making some efforts to answer that question in HCC right now, but I think for cholangio, that's still an open question. We did actually discuss a little bit about the circulating tumor DNA. We think that assay definitely has the advantage, perhaps capturing the heterogeneity more than just a post-treatment one biopsy that may only represent what you see in that biopsy. So the circulating tumor DNA definitely has the advantage, capturing some heterogeneity coming from all regions. Yeah.
So the reason the question was because we are doing the CTC, and when we do the sequencing, the allele frequency is 0.03%. And when we do in a biopsy or in a tissue, it could be 60%. So then it's kind of hard to believe if that 0.03% is the same mutation which we see in a tissue or in a CTC, is it real or not?
Right. So sounds like we have a lot of potential to follow up
Okay,
there.
thank you.
Thank you so much for your interest and questions, and let's have a break, very short break, 10:20, reconvene. We've got great stuff on deck.